Okay, so today we're going to be talking about proteomic methodologies and different assays. Can I just get a show of hands who does proteomics here? Yeah? Yeah? Alright, a few? Okay, two. Okay, well hopefully this is a nice introduction to the kind of quantitative assays you can do with proteomics. I always have this disclaimer that this is not a comprehensive suite of everything because I've tried to stick to mainly MS-based proteomics, but of course we've talked about immunoassays. I haven't gone into Western blots. I haven't gone into protein microarrays. Those do exist. We can talk about them more. If you guys want to have a lecture on it, you can always have one. But I think in general, proteomics is really moving towards shotgun-based, whole proteomic quantitative analysis. That's really what most of us will end up doing. I think that's what most of the papers end up doing. And when we do the case studies much later on, I'll try and include, someone suggested a really good plant proteomic study that was done with SRM. I don't know if it's anyone in this room, or if it was one of the seniors, but we'll go through that and we'll just discuss really how these proteomic studies hold up compared to other biomarker assays. Like I said, I have gone a bit backwards. The next lecture we'll have with me will be on gene expression studies. So we're not really following the traditional central dogma, but I'm sure you guys don't have any problem with that. Next week will be a lecture with Dave, and it's machine learning and RSD analysis. Good luck. I looked at the slides and they're terrifying. So hopefully mine is not too terrible. So why do I have a problem changing that? Okay, so just to recap our biomarkers, we're interested in these measurable entities that can provide diagnostic, prognostic, and treatment-orientated care. Ideally, we want them to have good sensitivity, specificity, and have good clinical application, that they can be measured in the clinic. But we need to go through discovery-based assays and really quite long-winded experiments to try and find out anything worthwhile to look for. So the main technologies, and these are the main ones, there are others, and I'm not even talking about metabolomics or lipidomics, which is a really big avenue of research now at the moment. But we're looking about proteomics, transcriptomics, and genomics mainly. Of course, there are others. So why are proteins, why is it good to study proteins over looking at DNA or over looking at transcripts? Why is a protein, I mean, some of it is up here. Does anyone want to attempt this with the hints on the slides? Our proteins are responsible for the actual phenotype, for what we see. They're responsible for the cellular processes that happen in the cell. So even though you can find a DNA or even a transcript, your mRNA transcript, that doesn't necessarily mean or correlate with the final outcome in your cell. There's post-translational modifications, alternative splicing, transcriptional regulation that influences the outcome of these to what you see at the end in the cell. So not always is measuring an mRNA going to correlate to that protein expression in your cell. So proteins really are the best marker for what is actually going on in your cell, specifically in diseases. They're the effector molecules, why enzymes, all of that, they're proteins. So proteomics as a biomarker discovery tool is really powerful, and you can really say a lot about what is happening from a functional perspective, from a disease perspective, if you study the proteins. And I included this picture, which is a very nice cross-section of a floral stem, which really shows the proteins are very dynamic molecules. They respond in both a temporal and a local fashion. So they move and they come up over time, and they go down, and there's a very dynamic expression of proteins, whereas DNA is there. Well, that's an advantage as well, right? Proteins, maybe you're not measuring it at the right time, and that's why you're not seeing it. But they're really much more dynamic, and in terms of looking at it, you're looking at it post-genomics. So you can look at isoforms, you can look at post-translational modifications, you can also look at interacting proteins, and that can tell you a lot from a functional point of view about what is happening in the disease, what's happening in my organism. So proteomics in general is defined as the large-scale detection, identification, quantification, and characterization. So you're really looking at quite a lot of things when you're identifying proteins in your cells. So just to recap the proteomic pipeline, we've been talking about that you're really starting with lots of molecules and really narrowing down into a few candidates that are exciting. So usually, sorry, there's no chairs. I don't know if you guys want to this one. Usually in the biomarker pipeline for proteomics, you will have a discovery proteomics and you'll have a targeted proteomics. You will usually have two phases of discovery. So in your discovery proteomics phase, you usually don't have prior knowledge, unless you're doing more of a targeted assay, but usually you're going based on, I don't know what proteins are going to be expressed in this disease. You may have an idea based on other studies that have been done, but you're not going in with any sort of hypothesis. Well, your hypothesis might be that proteins are changing, that's it. And what you're trying to do in your discovery proteomics is to optimize the number of IDs, good quality IDs that you can get in your MS experiment. That is usually what's happening in discovery. So you're going to spend much more time per sample to really optimize the number of proteins that you see and make sure that you're getting good quality spectra out of your MS, so that you have very good identification and hopefully good quantification, so that you can look at differential protein expression. So here, because you're spending more time per your sample, you will sacrifice how many samples you can analyze. So usually here, it's a much smaller cohort, and depending on your labeling or label-free strategy, you're not going to be able to analyze that many samples at a time. And you will usually use a high-resolution instrument that is able to detect minute changes in your MZ differences, because you want to ID a lot and get a lot of very good quantification information here in your unbiased discovery, which is also known as, well, we'll talk about shotgun proteomics in a minute. In your targeted proteomics, so after you've discovered these interesting biomarkers that you think may be worthwhile pursuing, you'll move on to your verification and your validation, where now you're looking at a discrete subset of peptides, which will translate to proteins. So you limit the number of features, so you're not going to spend so much time on your sample trying to get the best separation on your chromatography and analyze every single protein. You have decided, I'm going to look at these 10 proteins, and you're going to optimize your detection strategy for those. So you're optimizing here the highest sensitivity. So usually in your targeted proteomics, hopefully, if your PI has a lot of money, or you have an analyst that's very nice, you will analyze thousands of samples. So you can really validate very strongly the presence of your biomarkers. And usually here you're using a triple quad or an iTRAQ instrument that can spend a lot more time. You can do SRM assays with these instruments. Okay, so that's the differences between these two. Most of the time, most of us will spend doing discovery proteomics. We'll find some assays. We'll maybe validate one or two with Western blot, and then your honors or your MSc is finished. Or you've got a discovery cohort, and you're now validating those systems with targeted proteomics. So there's a number of methods that you can use, and I'm really focusing here on comparative proteomics, right? Like a disease state versus undiseased, or stress versus non-stressed, or acid stress, something like that. So you're looking at proteomic changes that differentiate the normal condition versus your normal state versus your condition, and that could be anything, right? And the proteome is very complex and it's very diverse. There are thousands and thousands and thousands and thousands of proteins in your sample. So usually you need some sort of separation strategy to try and tease out what you're seeing on the MS. And we'll talk about how to separate these proteins. So I'm focusing on quantitative methods. So you can do it's called single protein analysis on a 2D gel or a DIGE. I think some people are doing 2Ds, right? There was someone who mentioned 2Ds. Well, we'll talk about that. So you can either do a 2D page or DIGE, and this is kind of the workflow for it. I'm sorry, the colors are not resolving very well, but it will come up on the slides hopefully. And here, you look for differences in gel spots appearing or disappearing or having different abundances, and you can pick these spots and put them through the mass spec to analyze which proteins are coming up. You can do a shotgun analysis or discovery-based analysis where you're using very high throughput. With the MS, of course you use MS for 2D page to analyze your protein spots, but here you're looking at the entire proteome. And you can do either a labeled experiment, and there are different labeling strategies you can use, or you will do a label-free experiment where you're counting spectra or looking at your peak intensity. And we'll talk about all of those methods that you can use. Finally, in your quantitative method is single reaction monitoring, or also multiple reaction monitoring, MRM assays that are targeted. Of course, you can also validate your proteins with Western blot, with your immunoassays, with Illuminex, whatever strategy you want. I just try to focus in on these because we don't want to be here all day talking about every single proteomics you can do. If you guys want to talk about a specific experiment, like, we can definitely discuss it. So the benefits of proteomics, and especially quantitative proteomics, is that you have a simultaneously discovery, but also quantitation. So that's very powerful. So you know that this protein is expressed, but you also know how much of it is expressed, and is it differentially regulated compared to your normal or your healthy or whatever it is that you want to look at. So in your two-dimensional gel electrophoresis, basically you will have your protein mixture and you can separate it based on its isoelectric point. So that's according to your pH, and when the protein reaches a neutral pH it will stop. And so you separate it by first charge. You can use the pH strips either 4 to 7, there are 3 to 10, you get a variety of pH strips. You need to have some sort of understanding about what proteins are, what kind of separation you want. There are different reasons for using different ones. Then you take that strip, you turn it the other way, and then you can separate out by size. So instead of having, the single band that you guys might be used to, where in a band you'll have multiple proteins, here you can start getting very nice separations. And with computational analysis, you can compare. So this is the example of what a normal 2D protein looks like. I wish I could actually tell you what this is. I think this is a plant proteome, but I can't remember what I ended up getting. I looked at so many images. But now you can start looking at all these different proteins, and with computational analysis, when you look at disease versus normal, here you can see in the disease this very nice expression of a protein coming up that's not present in the normal condition. So now you can go and select the spots, do an in-gel digest, run it on the MS, and actually identify what that is. And so what's nice about this is you have actually quite good resolution on the gel. Right, but there's some proteins that are not going to be separated out on here, so you have to always keep that in mind. But 2D gels have been used very routinely and actually have been very good for identifying good protein changes. The problem with 2Ds, and if anyone has done a 2D, I did it on my master's and I made 106 gels. Because you need so many replicates to make sure what you're seeing is true, there's so much variability in a protein gel, it's very, very laborious. And then you have to individually pick spots, digest them, and then run them on the MS. So you can only really do, you could probably do a lot of protein spots if you really were dedicated, but there are problems with reproducibility with 2D page, traditional 2D page. Another problem is the limit of detection of your stain. So most of us would use Comasi stain, but Comasi actually doesn't have very good lower limits. So you can do like a colloidal stain or silver staining that gets you better resolution or you better sensitivity, but you are limited by what your stain can stain, right? And you need a lot of protein to run a 2D, usually. You're not going to be able to just analyze on the MS. MS, you don't need a lot of sample, obviously. So they actually came up with this technique called difference gel electrophoresis. So here you can do up to 3 samples, and you can label each individual samples with these spectrally resolvable dyes, which means these fluorescent dyes will come up in different absorbances, right? So they have their own profile. So what you do is you individually label them, pool them, and then when you run them, when you do your image analysis, then you can see and spectrally resolve this one's from that sample, this one's from that sample, this one's from that sample, and see which ones come up and down. So this is quite nice because you can actually pool and you only run a single one. You have less problems with variability. But you need fluorescent dyes, it's more expensive. Is it more expensive than running 100 gels? Probably not. So that's 2D page, which is used very routinely, but we are moving through high throughput now shotgun experiments. So mass specs have become really, really advanced instruments now. We can resolve things very well. They're extremely sensitive, very, very good definitions of mass accuracy on your MS. And so they've allowed us to investigate entire proteomes fairly quickly. So the nice thing about shotgun MS is you don't need that much sample. You can do quite a nice run with a small amount of sample, depending on the level of manipulation that you need. If you're going to do a quantitative label experiment, that's going to get more tricky. So you can high throughput ID thousands of proteins and quantify them at the same time. We can do label versus label free, and then of course relative versus absolute quantification. If you speak to Dave, he'll tell you there is no such thing as absolute quantification. It will always be relative to whatever you've spiked in. Usually with your absolute, you do a spike in standard, and so according to that, you can enumerate how much protein you have in your own sample. That is also actually a form of relative quantification, but we're splitting hairs now, so let's not go into that. So these are kind of the different strategies, but we're going to go into the different types of labels and things like that. So with label quantitative proteomics, you can compare protein abundances based on the presence of labels that you have tagged onto your protein, and there are a number of labeling options. You can do chemical labeling, or most of them are based on isotopic labeling. So you can do iCAT. iCAT is maybe an older methodology. I don't know if anyone still uses. I've never used iCAT. I think with iCAT you can only compare 2 samples at a time. You can do iTRAQ, isobaric tagging for relative and absolute quantification. That's what I used in my PhD, and you can use a tandem mass tag. There are other types of tags. These are the more commonly used ones. I'm trying to keep it simple on the slides. You can do a metabolic labeling with SILAC, which incorporates heavy versus light chains of amino acids. But here you need live cells. But metabolic labeling is very sensitive. Your cells take that up really nicely. So that's a very powerful approach. You can also do enzymatic labeling. I've never done that or seen that much work done on that, but either using deuterium or heavy oxygen. And the point of labeling is that if you have your different samples, so this is just a 4-plex, we're comparing 4 at a time, you take out your protein, and then you trypsin digest, and then, so this is an iTRAQ label, 114, 115, 116, 117. They go up to 8-plex. And you label every single peptide. And this one, it gets 114, this one 115, this one 116, 117. And then these get pooled. And what the tag does is it gives all those peptides the same mass in your first MS. So on the first MS, they're all going to fractionate in the same way. They're all going to get seen in the same way and pass into this tandem MS. On your tandem MS, when you fragment, your reporters come off, and they have different masses. So depending on the abundances of each reporter, you can see that these two samples had much more of this protein in that sample. So the advantage, so I don't want to get into the technicalities of the labeling technology. We can do a whole essay, a whole lecture on that. But the advantage of label experiments, what I want you guys to take home is that you can pool your samples together and do single runs on the MS. So you can multiplex. TMT does up to 10-plex now. iTRAQ does 8-plex, and they label in different ways, so you might choose based on how it labels the primary amines. And you can, by multiplexing and running into a single shot, you reduce your variation between your MS runs. If you guys have worked on the MS, you know that like the MS can be temperamental, your chromatography can change day to day. So if you're doing a label-free experiment, which we'll talk about, and you have to run each of those samples individually, you start getting more variation, which you have to then correct for later on. There is variation in quantitative experiments as well, and that's a whole other kettle of fish that we're going to talk about. But one of the points is that you get a reduced cost of your MS run, because you can just theoretically just do a single injection. Of course, you're probably going to fractionate that sample and run it over 10 fractions, but you don't have to run 10 individual samples. But they're much more expensive, these reagents. Usually your labels are expensive, and there's a lot more sample processing that you need to do, because you have to label and you have to make sure that you're combining in equal fractions, and that can introduce variability at each point. There are things you can do to correct for that and normalize for that, and there are a lot of pipelines that help you with setting up your quantitative experiment in the best statistical way that you know what you see is true. Are there any questions on this? So, moving on, label-free quantitative proteomics. It's more straightforward, and I put this in inverted commas because I think if anyone's done label-free, they know that it's not that straightforward to interpret. Like, you really do need to understand and know what you're doing. And it is more inexpensive from a point of view that you don't have to buy labels, and you don't have to have all these kits that come and that are very expensive. You don't have to manipulate your sample as much, you don't have to label and make sure that you're combining them in equal fractions, where you're introducing variability at each point. Here, in theory, if you have a lot of money and a lot of time, you can compare unlimited amount of samples. Obviously your label, you're limited, the most you can do is a 10-plex. There are people that run like multiple 10-plexes and they combine that. That has its own problems as well and becomes very tricky. So in theory, if you have a lot of samples, you want to go for a label-free approach. There's no multiplexing, which means you have to run them individually. So you can either do a spectral count, so based on the number of spectra. Did Dave give you an SRM lecture? I think he did. Maybe once upon a time. He sent me slides and he said he presented at UWC, so I haven't really gone into, he did SRM and apparently label-free approaches. Spectral counting, okay? No? Alright. He might do one at some point, really detailing this. I am really scratching the surface of how you do this. But you can do spectral count, or you can do this based on ion intensity or the peak area. Dave prefers spectral counting, and there are differences between the two, but this seems to be more sensitive and specific and less prone to error. So the problem with not being able to multiplex is you have to run individual samples, so you might have issues with batch run variability, where the MS is not behaving, say, the same way, or you're not getting the same separation as you did in your first run, your second run, your third run. But there are ways to normalize data. Bioinformatics is a very powerful tool that you can deal with these kind of issues. But always remember that these could be issues that you have. So if you have a small amount of samples you want to analyze, maybe go for a quantitative labeled approach. So what do I choose? I've given you guys all these options and everything, and it's like, it's so overwhelming. And I think Igon's in this boat now where he's having to decide, like, how am I going to do this? So there's no one size fits all, obviously. Each strategy has its own pros and cons. On the next slide, I've given you a really nice table that I found that details advantages and disadvantages of all the methods, so you can make a decision. But you need to decide, what do I want, what do I need, and what do I need to analyze. So what is my sample type? Sometimes the sample type is what limits you. Maybe, for example, Hygon he works on saliva. Saliva notoriously has a lot of amylase. It's just chock full. In fact, no one likes to do proteomics on saliva. I don't know if his supervisor hates him or what, but salivary proteomics is notoriously difficult. So he is having to look at maybe just separating out on the gel so you can cut out that amylase band and then deal with the rest of the sample. How complex is my sample? That's important. You might be limited if you've got a very highly complex sample. There are different strategies you need to employ. How many samples do I want to compare? That's what we've talked about. If you've only got 4, go for a 4-plex. What is the dynamic range? So can the labels do that? Labels do have influences on that. What instrument is available? So that's often what we end up making our decisions on, right? I only have access to this instrument, so therefore I can only run this. I can't do an MS3 experiment. Also funds. At the end of the day, that's what limits a lot of us, right? If like your supervisor doesn't have a lot of money, or if you don't have a big grant, you're not going to be able to do the sexiest experiment you were planning on doing with the coolest strategy. So this is a table which is a bit busy and probably not the best resolution, but here are the advantages and disadvantages between the gel-based methods and your gel-free methods. So here there's a bit more. There's the ICPL, and then we're talking about SRM later on. But I hope that this can help you and really be a summarized table that will help you decide if you want to do a proteomics experiment, which strategy you want to go for. So why can't I see anything? I've designed this experiment, I've done my quantitative thing, and I run it on the MS and I see nothing. Or I just see a whole bunch of the same protein. There are a lot of proteins in your biofluids, right? It's chock full of stuff. But most of the time, your interesting, and I put that in brackets because this is not always true, but your interesting biomarkers that you want to look at are not present at very high concentrations. And they end up getting masked by very abundant proteins. All of the biofluids or tissue samples that you can work on have abundant proteins that are going to mask it. So I just took an example here. In your serum, which is a very popular biofluid, 98% of your proteome is highly abundant proteins. 98. I mean, that's huge. That's most of the MS we'll see is albumin and IgG. That's it. And a few others, like actin and things like that. Heart tissue, 40% of the proteome is myosin and actin. And so this ends up masking, because the MS can only do so much, right? It can only see so many events at a time. If it's spending all of its time looking at these proteins, all your low abundant proteins are going to go undetected. And a lot of the time, this is one of the biggest problems of proteomic experiments, where people don't think about these abundant proteins and getting rid of them. So you need to reduce the complexity. You can do a depletion experiment where you just deplete out your proteins. There are columns you can bind that bind these proteins and deplete them out, and then what you're left with is a less complex sample. You can also enrich for your proteins, right? So that's more like you have to know what you're looking for and just to try and wash off the rest of these proteins. Fractionation is, you'll always do a fractionation at some point, either on a C18 column, or if you can do HPLC, or there's some form of fractionation, off-gel fractionation. You can fractionate on a gel. There are different fractionation strategies as well. But you need to do these steps to reduce the complexity, otherwise you're going to have a very tough time to find anything very interesting. With these strategies, there are problems of you're getting like these kind of off-target effects. So especially if you're depleting, you're hoping that you're just binding albumin and IgG, but there's going to be other proteins that stick and that might also get depleted along the way. So you need to be aware of that. You might end up depleting other things. And that's just That's just something that happens, and you have to just try and hopefully your depletion strategy had introduced the same error for all the samples. So really the key of proteomics experiments is very consistent and careful sample preparation. You need to be very consistent, very careful, monitoring what you're doing all along the way, trying to reduce the error that you see. Because there's so much sample manipulation that happens, all along the way things happen and can introduce this variation. And you need to try and bioinformatically think of how you can correct for this, how you can normalize your data. You have proteins that act as your housekeeping proteins that you can use to normalize your data. People normalize on total protein abundance as well. That's a strategy. I really encourage you guys to read as much as you can around what people have done in your field, but also in other fields, because there's some very clever strategies to get around these issues. So bioinformatics is probably, I think, the toughest part of a proteomics experiment, and probably for other quantitative experiments, because there are so many different ways of analyzing your data. I would really recommend that you decide your strategy before you start. There are so many people that come after the fact, and they're like, I did an iTRAQ and I wanted to look at this, and they had no plan to reduce complexity. They have all this info that they want to analyze, and they have no idea how to analyze. So try and speak to a bioinformatician. You know, it's hard to become an expert in molecular biology and bioinformatics and biostatistics. Talk to maybe someone like Dave, he's a really good resource, to decide on a good strategy. Think about what the issues are with your experiment. Is the quantitative method I have chosen, my label, going to introduce variation? Is my label-free approach, did I have issues with MS on that day? There are a number of things like noise filtering and centroiding strategies that you need to use to deal with your proteomics data. Also, going from peptides to proteins, do I have a database? Probably not if I'm not working on humans. I mean, there are Arabidopsis and that kind of thing, you can get a database for it. But a lot of us work on organisms that don't have available databases for our organisms. So you have to use a for example, when I was working on abalone, I had to use an all-molluscan database. So that can introduce false discovery when you're not using a database that is specific to your organism. So that's something to think about. And minimum 2 unique peptides, that is the norm, right? And for identifying a peptide, usually 2 or more. Identifying on 1 unique peptide, it can be done, but you need to be very confident about what you're looking at. So if those of you who have not heard of it, the transproteomic pipeline, it's a very useful bioinformatic tool that you can use that allow you to move through the bioinformatic pipeline with tools that they suggest. There are a lot of publications on it. This is one that you can look at if you want, Bioinformatics for Omics Data. Really suggest you look at it. They detail and they have a really nice tutorial going through each step and which one you should go for. I also put here what fold change. What is a significant fold change? And we talk about this all the time. A lot of people toss out anything that is not more than 2-fold. Or you have to look at, I'm only going to look at 10-fold fold change. But what is a biologically significant fold change? I don't know. I don't actually have that answer, because sometimes there are minute differences that you can detect in a cell that are biologically significant, but we end up tossing out anything that isn't more than 2-fold change. Is that a mistake? I don't know. What some people do is they stratify their data. So first they look at all the fold change, and then they get stricter and stricter and stricter to try and narrow in on what's important. But I don't have an answer for this. We talk about it at length, always in meetings. What is a significant fold change? I don't know if anyone has any points. Ashwell, maybe you want to tell us what you guys do?
We stick to 2 and 3. 3 maximum, 3.
2 minimum.
Yeah, we go 2 minimum, but yeah, 3.
That's the standard, yeah. But it is this question of are we omitting things that are maybe interesting? But how statistically valid are they? Yeah. So it's a tricky question, and I think we have to stick with maybe the norms, but think about perhaps you're omitting things that could be interesting. So for validation, we use targeted assays. Now you've come up with your candidate protein list, you've done your statistical analysis, you have these fold changes that look really good, you've got your list of, say, 10. You need to now do a targeted proteomics. So there are immunoassays and protein microarrays. I'm not really talking about them today. They are there, very powerful Western blots. That's how I validated all of my proteins. If you have antibodies, we already talked about that. Most people now are starting to move to selected reaction monitoring. It's also known as multiple reaction monitoring. So SRM, MRM, it's an absolute quant. Again, you use synthetic peptides for your target. You have to generate synthetic peptides. That's one of the difficulties with SRM assays. You spike them in, there are standards, and then you can make a calibration concentration curve. And then based on your synthetic peptides, you can enumerate what the absolute amount of your target peptide you have. With SRM assays, there is a lot of optimization you need to do. It is not simple. I think a lot of people think, I'm just going to do an SRM, it's so quick, so easy, my analyst can do it all for me. No, you have to optimize. But they were extremely powerful. SRM assays are extremely sensitive, highly, highly reproducible. Once you've optimized your SRM assay, you're good to go. You can analyze thousands of samples in different tissues because now you know which your target peptide is. So it's very easy. Easy. So it's reliable quantification of analytes in low abundance, complex mixtures, so you probably don't need to do a lot of fractionation because you're asking the MS to filter things out for you. You don't need to do that much fractionation. So usually, as I said, it's highly specific, sensitive, and very throughput. These are some nice reviews that I found if you guys want to have a read through these papers. They're really detailed SRM assays. I'm not going into that much detail today. But usually you use a triple quadrupole, which I think you guys have a triple quad, right? No? You don't know. Okay, you use a triple quadrupole, and the first and third quadrupole, they act as filters for your specific MZ. And the transition, or this filtering that happens is known as a transition. You'll often hear that word that comes up, how many transitions. So you need previous information, right? Usually you can get it from your shotgun proteomics that you did. You have some spectra for your peptide or your protein of interest. And there are a lot of repositories. If you don't have information, you want to set up an SRM assay, there are tons of repositories. I will make a little note of all the, there's SRM Atlas, Peptide Atlas, a whole bunch of repositories that have information so that you need to decide on your target protein, which unique peptide you want to identify it based on, and then its fragment ion. It needs to have good intensity. Because as you're moving through the quadrupoles, right, you're choosing your precursor ion here, but here you've had, you're interested in the green, but the blue and the green have come together, so your green is in here. You fragment on your second quadrupole, and then in your third quadrupole, you're just selecting your fragment ion. And that's here when you can be very selective, and only your green ones come through. So you need previous information, you need to optimize it, you need synthetic peptides so that you can get good spectral information so that you can design these experiments. It's also good, it's not always done, but to have a housekeeping protein just so that you can normalize your data and making sure when you're quantifying you're getting statistically valid quantification. So choosing housekeeping proteins obviously depends on your organism, your tissue, and what's being expressed. We can do a whole SRM lecture if you guys really want SRM techniques. This is really scratching the surface of how you would do an SRM experiment. So how do you make the most of your proteomic experiments? Because it's a really powerful tool, but things can go wrong. So you want to maximize the likelihood of identifying your protein with real potential as a disease-associated biomarker. It's all good and well to measure proteins, but if they're not part of your disease functional pathways, it's not very helpful. So you need these two phases. We've talked about discovery and validation. I've included these 3 papers here, but there are tons of papers that talk about why biomarker discovery in proteomics has failed so far. It has not been very good. And really, it's because there are a lot of errors that happen based on ill design of your study, or you didn't have the right cohort or whatever. So you need to clearly define your research question early on. Often you're like, oh, I have access to the sample type, I'll just analyze by proteomics. What are you trying to answer? What are you going to look for? You need to choose the appropriate sample type. I think here it's very important to talk to your mass spec analyst. Your analyst has a lot of knowledge, he has run a lot of different samples. He will know what problems there are. Probably early on he will say, this is going to be a problem, this is going to be a problem. So collaborate with your analyst, with your bioinformatician if you have access to one, and anyone else in the field who's done a proteomic experiment. Message them, email them, say, I'm looking to do this, like, what have been your experiences? Honestly, ResearchGate is a really good tool. You can talk to other scientists who are doing similar experiments and say, what was the problem? I'm seeing this, I'm not getting a good protein count, why? So I really talk to people before you run off and start designing your proteomic study. It will really help. Have protocols in place. If you are not in charge of sample acquisition, things go bad. Especially true in the clinic, you have different nurses, different clinics, different recruiters, the patients, and the samples are not in your, if they're not in your hands, you cannot control that things aren't being done slightly differently here, and then all you're measuring are differences in your acquisition. So if you cannot control your sample acquisition, make sure you have done protocols, standard operating protocols that can be followed. Identify your variables early on. There's variables introduced because of your labeling strategy, because of the different sample types. I reviewed a paper that was looking at, they were like heat stressing these little mollusks. And so they basically dried out this whole one mollusk and then compared that to just a normal mollusk. But they were also interested in like the rehydration of these mollusks. So instead of having a control that was never dried out, I said they had already done the analysis and they didn't really find, well they found things but it didn't seem to correlate at all. And I told them they should have looked at dried out and rehydrated versus the completely dried out. That would have been a better control. So try and think about these kind of variables. As I've said, have your bioinformatics strategy before you begin. That's really important so that you know what you're doing downstream. It's so hard to deal with your proteomics data. If you know that you're like, I'm going to follow this strategy, I'm going to filter out my proteins like this, it just makes for a much better time, honestly. Right, so the take-home messages: proteomics, they allow large-scale, high-throughput analyses of your complex biological system. Very powerful strategy. It's probably my favorite omics to deal with. You allow biologically relevant targeting of disease diagnosis, prognosis, and drug response evaluations, hopefully, if you're lucky. There are numerous approaches that can be used for quantitative experiments, and you should think about which advantage or disadvantage you're willing to deal with. You need to do some sort of pre-fractionation and depletion to reduce the sample complexity most of the time. Sometimes your sample's not that complex, so then you're good to go. That's really lucky. Issues including reproducibility of your experimental setup, your detection limits, and development of appropriate statistical approaches need to be taken into account. Statistically, how am I going to deal with this? I am not a statistical expert. I find statistics really hard. And proteomics really needs strong, robust statistics. So talk to a statistician. There's nothing wrong with that. Yeah, it's a very complex, multi-step process, and you need to report this in your publication. There's now the MIPI, Minimum Information About Proteomic Experiment. Have a look at the MIPI guidelines. It will help you design your experiments. Your validation is a key step in biomarker discovery. You need to validate. What you see in the quantitative experiment is not necessarily true. You need to validate using a different technique, or an SRM, or an immunoassay, or something, to validate a select handful of your proteins to say, what I've seen is true. This protein does come up, or it does disappear in my condition. Validation is very important. I've included these. There are tons of publications to read on the topic if you're interested. I think these are quite nice in terms of biomarker discovery. They really delve into the topic really nicely. All the problems, everything people have dealt with. Have a read through some of these if you're interested. And yeah, don't hesitate to contact me. I mean, you guys have experts here as well, but it's always good to chat to other people and just discuss what you guys want to do. Are there any questions? Do you guys want to do proteomics now?